Restriction enzymes. Definition. Restriction enzymes, also called restriction endonuclease, is a bacterial enzyme that cleaves the double-stranded DNA at specific sequences. Each restriction enzymes act on its own specific sites. Otherwise, we are calling it as a recognizing sites or cutting sites. This recognizing site contains 4 to 8 base pairs of nucleotide sequences. These nucleotide base pair sequences are recognized by restriction enzyme. Restriction enzyme. Restriction enzyme are otherwise called as molecular scissors. It is a nuclease enzyme. It consists of protein that recognizes a specific sequence of DNA bases and cuts in its particular subunit. This enzyme is produced in bacteria. Bacterium is made up of single cell, some of which are pathogenic, which infect mammals and cause diseases. Bacteriophage are viruses that specifically infect bacteria, and it kills the bacterium during the process of producing more bacteriophage. Restriction endonuclease. Bacteria use restriction enzymes to kill viruses. These enzymes attack the viral DNA and break it into useless fragments. These fragments become inactive. Sometimes it is digested. Some strains of bacteria are immune to bacteriophage infection. It is called as host-controlled restriction. Restriction occurs in the bacterium, produces an restriction endonuclease that degrades the phage DNA before it replicates. Restriction enzyme action. The phage DNA is entered into the bacterium. Once it entered into the bacterium, the restriction endonuclease present in the bacterium cleaves the DNA, that is phage DNA, and it becomes inactive. The bacterial DNA is not cleaved because the bacterial DNA is methylated. The recognizing sequence of the bacterial DNA is methylated. So, the restriction endonuclease are unable to bind the recognizing sequence. Recognition site palindromic sequence. Restriction enzyme comes into contact with a DNA specific sequence with a shape that matches a part of the enzyme called recognition site. It wraps around the DNA and causes a break in both strands of the DNA molecule. Recognition sites are usually short. It contains 4 to 8 nucleotides. This recognition site will have the palindromic sequence. Is a sequence. It is made up of nucleic acids. That is a mirror image of the same. When it reads from 5' to 3' direction. Here they are given one example. GAATTC. It is arranged in 5' to 3' direction. CTTA AAG. It is arranged in 3' to 5' direction. Both the faces are mirror image of each other. Blunt ends and sticky ends. Restriction enzymes recognize a specific sequence of nucleotides and produce a double-stranded cut in the DNA. These cuts are of two types, blunt end, sticky end or cohesive ends. The examples are given here. The blunt end may produce an equal cut. The sticky end may produce an unequal cut. Blunt ends and sticky ends. Many restriction endonuclease do a simple double stranded cut in the middle of the recognition sequence and produce the blunt or flush ends. These blunt-ended fragments can be joined to another DNA fragments with blunt ends. Blunt end is of considerable importance in the design of a gene cloning experiment. A large number of restriction endonuclease cut DNA in a slightly different way, that is, not cut at exactly the same position and produce the sticky end or cohesive end. DNA fragments with complementary sticky ends can be combined to create new molecules which allow the creation and manipulation of DNA sequences from different sources. One important feature of sticky end is that restriction endonuclease with different recognition sequence may produce the same sticky ends. Example, BOM H1 recognition sequence GGATCC, BGL2 recognition sequence AGATCT. Both produce GATC sticky ends. BOM H1 restriction enzyme is derived from Bacillus amyloliquefaciens. BGL2 restriction enzyme derived from the bacteria Bacillus globigii. Fragments of DNA produced by cleavage with either of these enzymes can be joined to each other if it carry a complementary sticky ends. The cleaved sticky end may join and produce a new fragment. One of the example given here, EcoR1 makes two different sticky ends. The two different sticky ends may combine together and may produce a new fragment. Recognition sequence calculation. Recognition sequence for a particular restriction endonuclease in a DNA molecule of known length can be calculated mathematically. Example, tetranucleotide sequence GATC may present the sequence again after 256 nucleotide bases. Per power 4 means 256 nucleotide. The same manner, hexanucleotide may present the sequence again after 4096 nucleotide bases. GC, the nitrogenous base, present 50% in nucleotide. Nomenclature of restriction enzymes. The restriction enzyme first letter denotes its genus name. Second two letters denotes its species name. Fourth letter denotes its strain. Fifth letter denotes its order isolated. Here they are given two different examples. EcoR1. Genus name is Escherichia. Species name is coli. Strain R. Order isolated first order. Recognition sequence or recognition site GAATTC. End produced is sticky end. Hint 3. Haemophilus influenzae. Strain is D. Order isolated 3. Recognition site AAGCTT. It produces sticky end. Type 1 restriction enzymes. Type 1 restriction enzymes are complex and have multi-subunit. It may have both endonuclease and methylase activities. They require ATP, Mg2 plus cofactor and S-adenosyl L-methionine, which is required for cleavage. They are single multifunctional enzymes which are capable of both restriction and modification activities. It recognizes up to 15 base pair in length and cleavage site is 1000 base pair. They show specificity for recognition site but not for cleavage. They produce heterogeneous type of fragments. So mostly it is not used in gene cloning techniques. Example, EcoB and EcoK. Example of restriction enzymes. EcoR1 restriction enzyme may cleave the restriction site in a specific manner and produce sticky ends. Type 2 restriction enzymes. Type 2 restriction enzymes are made up of single subunit cleave within or at short distance from DNA recognition sequence. They are simple enzymes having single polypeptide. They have separate methylase and endonuclease activity. It requires mostly Mg2 plus as cofactor. This enzyme performs only the restriction function. It is the most common restriction enzyme used in gene cloning techniques. The recognition and cutting site is the same for this enzyme. They generally recognize 6 nucleotides. Sometimes it recognizes 4, sometimes it recognizes 5, sometimes it recognizes 8 base pair also. Example, PUV1, PUV2, echo R1 cleave DNA to generate different ends. Type 3 restriction enzymes. Type 3 restriction enzymes are intermediate between type 1 and type 2 endonuclease. They cleave DNA in the immediate vicinity, that is nearby of the recognition site. Example, echo P1, HEN3, echo P15, etc. They cut DNA up to 20 to 30 base pairs away from the recognition site. These enzymes contain more than one subunit and requires S-adenosyl, L-methionine and ATP and Mg2 plus cofactors for their roles in DNA methylation and restriction enzymes. Restriction enzyme application. Restriction endonuclease are synthesized by bacteria and used for its own defense mechanism. Restriction enzyme recognize the source of incoming, that is foreign DNA, and destroy if it recognizes the incoming DNA as foreign DNA. Restriction endonuclease spot specific sequence in the incoming DNA and digest the DNA into fragments. They cut the DNA either at specific sites or more randomly. These DNA fragments are used in molecular cloning. They are used to digest DNA for gene analysis. Restriction enzymes are used in genetic engineering to cut the gene of interest from the source of DNA, that is specific gene, and to insert them into the plasmid vectors. It is used for construction of recombinant DNA and gene cloning experiments. Thank you.